talking about today, we will first discuss what Amplicon sequencing is, then how Amplicon library preparation is different from whole genome library preparation. We will cover which kits and workflows can be used to prepare Amplicon libraries. We will look at what an Amplicon sequencing run looks like. We'll discuss how low base diversity can affect a sequencing run. What's the best way to sequence low diversity Amplicon libraries? And what analyses are available for Amplicon data? First, what is Amplicon sequencing? Amplicon sequencing is a method of targeted sequencing to look at a specific locus, region, or set of regions. It lets us to get a closer look of specific genes or targets of interest. It allows for deeper sequencing of these regions, resulting in higher sensitivity, and it is more cost-effective and time-efficient than other types of sequencing, such as whole genome sequencing, when we are interested in only a specific region. Applications of Amplicon sequencing include cancer, forensics, inherited disease, agrigenomics, metagenomics, and more. Now, let's compare whole genome sequencing libraries with Amplicon libraries. First, we will look at a genomic DNA library preparation. We start with genomic DNA, and then we randomly fragment it into smaller pieces, creating many unique fragments, and then we add adapter sequences to create a library ready for sequencing. This results in a library with many different unique inserts, which yields a balanced or diverse library, because at any given position, we expect roughly equal proportions of A's, T's, C's, and G's. You can see at this single position, each base is present and balanced. Let's contrast this with an Amplicon library prep. In this method, we start with genomic DNA, and we selectively amplify a single region and then add adapters. This results in a library where the inserts are largely the same sequence, creating a very low base diversity library. You can see at a given position, only a single base may be represented. These examples represent the far ends of the spectrum, from highest to the lowest in diversity. You can imagine if we had a library composed of multiple amplicons, it would increase the overall diversity of the library, though it would still not be as diverse as a genomic library. And we will see an example of this a little later. Now, which kits or workflows can we use to prepare Amplicon libraries? We will discuss 3 methods today for Amplicon library generation, and after each, I will include some resources for more information. The first workflow is AmpliSeq for Illumina. AmpliSeq for Illumina allows for both custom and fixed panel options, and it is compatible with low input integrated samples. The second workflow we will discuss is Nextera XT and Nextera DNA Flex library preparations. For these, PCR amplicons are prepared in advance, and random fragmentation is used during the preparation, which adds base diversity to the libraries. The third workflow is the Illumina demonstrated 16S protocol. This is a 2-step PCR workflow. It has a single target region per library and can be customized for novel targets. Our first workflow is AmpliSeq for Illumina. We will start with our input material, which may be DNA or RNA or both, depending on the panel. Then, primer pairs will create multiple amplicons across the region or regions of interest. The primer sequences are removed, then we ligate adapters and amplify the library fragments with PCR. After a cleanup, the libraries are ready for QC and sequencing. When you are validating your AmpliSeq for Illumina libraries on the fragment analyzer, bioanalyzer, or other trace instrument, the distribution that you will see will depend on the panel content, and Amplicon size. These are two examples of AmpliSeq libraries. For ready-to-use panels, there will be an example trace in the reference guide for the expected library distribution. We currently have the option to pull up to 384 samples per run using index sets A through D. Panels contain multiple Amplicon targets, which add diversity to the libraries. There are DNA and RNA panel options. Input can range from 1 nanogram to 100 nanograms, and we typically recommend 10 nanograms of input material per pool for high-quality input. With lower quality samples, we may want to increase the input material, if possible. We have 12 ready-to-use panels for use with DNA input, RNA input, or both, depending on the panel, and we are continuing to build our AmpliSeq portfolio. We have additional community panels available, which have been created by key opinion leaders in their fields. Some examples of the available community panels include the Canadian Inherited Cancer Panel, Antimicrobial Research Resistance Panel, and Autism Research Panel. And we can create custom DNA AmpliSeq panels using Design Studio, targeting not only the human genome, but other species as well. I'll give a brief overview of the custom AmpliSeq design process. First, in Design Studio, we will start a new design. If we choose RNA as the assay type, then we will have three options. Expression, which is a single pool design for targeted gene expression analysis. Fusion, which is for targeted gene fusion panels. And TruSeq targeted RNA, which is a sequencing-based gene expression assay, which allows up to 1,000 targets in as many as 384 samples on the MiSeq. Please note that these options are only available for the human genome. If instead we choose DNA as the assay type, then we have four AmpliSeq options. Gene, which is useful for covering full genes or regions of interest. Hotspot, which allows for designing panels that target SNPs and small indels. On Demand, which allows you to select from predetermined wet lab tested primer pairs. And Enrichment, which allows for targeting large panels for use with the Nextera Rapid Capture Custom or Nextera Flex for Enrichment library prep kits. We can then choose the species we want the panel to target. We can then configure more specifics about the design, choosing the sample type and the Amplicon length. For regular input, we can choose one of 4 Amplicon lengths, whereas with FFPE and cfDNA, or cell-free DNA options, the Amplicon length options will be limited to smaller sizes to ensure good panel performance. Once we've configured the design parameters, we can add the targets we would like to cover by gene name, chromosomal coordinate, or we can use a template to upload multiple targets at once. We can also copy Amplicons from pre-existing designs or from community and Phix panels. The minimum number of Amplicons for design is 12, while the maximum is over 12,000. After all of the targets have been added, we submit the design. Once the design completes, we can review the coverage and further optimize the design if we would like. We have detailed recorded webinars that discuss AmpliSeq for Illumina and Design Studio. I've also included links for our ready-to-use AmpliSeq for Illumina panels, our community panels, and Design Studio for creating your own custom panel. Now, let's discuss how to use Nextera XT DNA Library Preparation Kit to prepare Amplicon libraries. First, you will need to design and create the amplicons you are interested in sequencing. Once we have PCR amplicons, we mix them with the Nextera transposomes. The transposomes tagment them to fragment and add tagging sequences to the ends of each fragment. We then use PCR to add the full adapter sequences and uniquely index each sample to complete the libraries. When checking Nextera XT libraries, we typically see a broad distribution of fragment sizes. The bioanalyzer traces here are from genomic DNA input. With Amplicon input, the input DNA will be smaller, so we can see smaller final library sizes and may see more narrow distributions. For Nextera XT library preparation, you will need 1 nanogram of input material, and this can be 1 amplicon or multiple amplicons. Amplicons will need to be 300 base pairs or larger. To ensure good coverage of the complete target region, we recommend adding an extra 50 base pairs to either side. Library preparation will require the Nextera XT library preparation kit and 1 or more of the Nextera XT indexing kits. With multiple indexing kits, we are able to pull up to 384 uniquely indexed libraries per sequencing run. And the random enzymatic fragmentation in the workflow adds base diversity to the final library. For more information about this workflow, you can refer to the support page. We also have a great recorded webinar available at the link here. The Nextera DNA Flex Library Preparation Kit is also an option for Amplicon library preparation. While the Nextera XT and Nextera DNA Flex workflows are similar, there are some key differences to consider. To prepare libraries with the Nextera DNA Flex Kit, we combine the DNA amplicons with bead-linked transposomes to tag and fragment the amplicons. We then finish the library preparation by uniquely indexing and completing the adapter sequences using PCR. This is the fragment analyzer trace for a Nextera DNA Flex library prepared with genomic DNA. The Nextera DNA Flex workflow does include a bead-based size selection step after the indexing PCR, so depending on the size of the input amplicons, you may need to adjust the final size selection, and this would lead to a shift in the distribution towards a smaller average size and base pairs. The Nextera DNA Flex Kit allows for flexibility for the amount of input material. Similar to Nextera XT, single or multiple amplicons can be used. Nextera Flex can be used with amplicons 150 base pairs and larger. As with Nextera XT, we recommend adding an additional 50 base pairs to either side of the region of interest. Library preparation requires a Nextera DNA Flex Library Preparation Kit and a Nextera DNA Combinatorial Dual or Unique Dual Index Kit. And we can pull up to 384 uniquely indexed libraries with the Nextera Unique Dual Indexes for sequencing. We also have a recorded webinar with useful information for the Nextera Flex workflow. The support page linked here includes protocol information and frequently asked questions. The final workflow we will be discussing today is the Illumina demonstrated Amplicon workflow, one application of which is the 16S metagenomics protocol. This is a two-step PCR workflow. In the first PCR, we use a locus-specific primers with overhangs to amplify a single region of interest. We then use a second PCR, which utilizes the overhangs to add the rest of the necessary adapter sequences and uniquely indexes the samples to complete the libraries. Then we are ready to QC the libraries and proceed to sequencing. When checking your libraries, you will see a discrete peak. As the libraries target a single amplicon, the distribution will typically be very narrow, but you can see variability if you are targeting a variable length locus or the same locus in a mixed species sample. For the demonstrated Amplicon workflow, each library targets a single region for amplification. It requires 12.5 nanograms of input and uses the Nextera XT indexing kits to allow for pooling up to 384 libraries per run. You can design your own locus-specific primers for the first PCR to target novel amplicons. This workflow is very similar to many published protocols for targeted amplicon sequencing. However, using this workflow ensures that your libraries will be compatible with any Illumina sequencer without any custom settings or sequencing primers. There are a few considerations for designing locus-specific primers. First, we should target an amplicon that will have about 50 base pairs of overlapping sequence when sequenced. For example, if running a 2x300 base pair paired end read, Illumina recommends having an insert size of 550 base pairs or smaller so that the bases sequence at the end of each read overlap. Second, the locus-specific portion of the primer must have a melting temperature of 60 to 65 degrees Celsius. For melting temperature calculation, only use the locus-specific portion of the primer. For hairpin and dimer calculations, use the full sequence including the overhang. Lastly, we recommend using standard desalting purification when ordering oligo primer sets. Here is a link to the Illumina 16S demonstrated protocol. This is a quick overview table of the 4 preparation methods that we've discussed as a reference. Now that we have discussed how to prepare libraries, let's proceed to sequencing, and we will take a look at what Amplicon sequencing runs look like. First, let's discuss 3 main metrics we will be considering to evaluate these sequencing runs. The first is % base. This will tell us the percentage of A's, T's, C's, and G's in each cycle of the sequencing run. The second metric is the % passing filter, which tells us the percentage of the clusters on the flow cell that are giving pure signal and that can be used to generate sequencing data. The third metric is the Q-score. We will be looking at the percentage of base calls above Q30, which is synonymous with a 99.9% confidence in the base call. The higher the percent at or above Q30, the higher the overall run quality. First, let's look at the run metrics for a balanced, diverse library. These are metrics from a Phix run, which is a viral genomic library. This is the percent base plot. On the x-axis, we have the cycle in the sequencing run, and on the y-axis, we have a percent scale. Each of the 4 bases is represented by a color, and each has its own line. When we look at the percent base, we see fairly equal representation of each base in each cycle. And the base percentages are even throughout the run, indicating that the bases are balanced. We can then look at the percent passing filter. The blue whisker box plot represents the raw cluster density. In this graph, we see is about 700K or 1,000 per millimeter squared. The green box plot indicates the cluster's passing filter. The green plot is very close to the blue plot, which indicates a high percentage of the raw clusters are also passing filter. For this run, the passing filter is just over 95%. Now let's look at the Q30 for the run to evaluate the quality. On the x-axis, we have the Q-score. The higher the Q-score, the higher the quality. On the y-axis, we have the number of reads in millions. A Q-score of 30 or above is considered high-quality data, and we can see that it is high for this run, around 90%. This next run is for an AmpliSeq for Illumina panel sequenced on the MiSeq using V3 chemistry. Each library includes approximately 4,600 different Amplicons. When we look at the percent base plot, we see that it is spiky, with one base having a higher percentage than the other bases at a given cycle. And this skew towards one base changes from cycle to cycle. Because the panel is made of a mixture of Amplicons, the overall diversity is much lower than the previous example of the randomly fragmented genomic DNA library of Phix. We can now look at the clusters passing filter. The green box, clusters passing filter, is quite close to the blue, which is the raw cluster density. The clusters passing filter for this run is about 82%. And last, we look at the quality for the run, and we see that our Q30 percentage is at 87.6%, which is quite high and tells us that the data quality is good. This last run is a 16S library preparation of an ATCC microbiome standard on the MiSeq using V3 chemistry. Each library is a single amplicon of the variable V3 and V4 regions of the 16S rRNA gene. Different bacterial species will have slightly different sequences for this region. When we look at the percent base plot, it is extremely spiky, with one base reaching as high as 90% of the signal at a given cycle. Such low diversity is typical of sequencing a single amplicon, which has very little variation between samples. Runs with such super low diversity samples can benefit from Phix spike-in to introduce base diversity. In fact, this run includes 10% Phix spike-in, which is why the maximum signal from a given base is around 90% rather than 100%. We can now look at the cluster's passing filter. This run had a raw cluster density of about 600K per millimeter squared, lower than the typical density of 1,200 to 1,400K per millimeter squared for V3 runs. This is to help mitigate the effects of the low diversity. Since there are fewer clusters that are well spaced apart, the cluster's passing filter is around 90% for this run. We will review this concept more later in the webinar. And last, we look at the quality for the run. We see that our Q30 percentage is at 70.1%, which is still quite high and tells us that the data quality is good. Now that we have seen what Amplicon sequencing runs look like, let's discuss how low diversity can impact a sequencing run. The first impact low diversity can have is on template generation steps. On non-patterned flow cells, during template generation, the sequencer creates a map of the position of each cluster on the flow cell. If there are multiple bases present, it is easy to distinguish each cluster and make an accurate map. With low diversity sequencing, at a given cycle, only a single base may be present, so clusters that are close together, showing the same base, may be seen by the sequencer as a single cluster, creating an inaccurate cluster map. The second impact is on matrix correction. Matrix correction is how the sequencer accounts for crosstalk between the fluorophores and corrects intensities. You can see with this emission plot that there is spectral overlap in the intensities that come from the bases. So we have to correct for this to confidently distinguish each base. If the majority of clusters are all showing the same base in a given cycle, the intensity baselines will be skewed, which will impact the matrix correction. If we have a poor map from template generation and inaccurate matrix correction, the sequencing software will not be able to make confident base calls, which will result in low data quality for the run. For more information about template generation, matrix correction, and other considerations for low diversity sequencing, please see the recorded webinar available at the link here. Now that we have discussed how low diversity can impact a run, let's cover the best way to sequence low diversity Amplicon samples. We want to mitigate the low diversity of libraries in order to improve data output and quality. If we have higher diversity Amplicon libraries, we may not need to use the upcoming strategies to improve the run. If you have any questions about the best way to sequence your Amplicon libraries, please feel free to reach out to technical support. The first way we can improve low diversity Amplicon sequencing is to spike in the well-balanced genomic library, Phix. For a well-balanced run, we recommend spiking in 1% of Phix as a sequencing control. For low diversity sequencing, we can also use Phix to add base diversity at each cycle. On the MiSeq using V3 chemistry, we recommend spiking in between 5 and 10% Phix into your library pool. We want at least 5% aligning during the run, so you may need to spike in a higher percentage to ensure that 5% aligns. Spiking in Phix will increase and improve the base diversity in the run, which will improve the Q-scores, so the quality, for the run, and will increase the percent of clusters passing filter, which corresponds to an increase in data output. Please note that if we are sequencing tagmented amplicons, we don't need to add as much Phix due to the random fragmentation of the transposomes. The Phix spike-in percentage will depend on the instrument you are using. We have a helpful bulletin which outlines the recommendations for each of our sequencers. The second way we can improve low diversity sequencing is by lowering cluster density. For example, for a well-balanced library sequenced on the MiSeq with V3 chemistry, we typically want to see a cluster density of 1200 to 1400 K per millimeter squared. For a low diversity run, we want to lower that to between 700 and 1000 K per millimeter squared. Cluster density may be adjusted in subsequent runs to maximize quality and data output. If we reduce cluster density, each cluster has more space, allowing for a more accurate map and sequencing. Lowering cluster density will not change the percent base, but it will improve the quality of the run and increase the cluster's passing filter, so it will increase the output of the run. Once we have our sequencing data, how can Amplicon data be analyzed? There are both BaseSpace and local analysis options available for Amplicon data sets. On BaseSpace, if our input is DNA, we can use the DNA Amplicon app. If our input is RNA, we can use the RNA Amplicon app. If our input is both DNA and RNA, instead of launching each app separately, we have a combined DNA plus RNA Amplicon app that will run both analyses. For local analysis, we can use Local Run Manager, or LRM, either the on or off instrument version. LRM is designed for benchtop sequencers, including the MiniSeq, MiSeq, NextSeq, and iSeq. It is not designed for the HiSeq series or NovaSeq. Similar to the BaseSpace apps, if we have DNA input, we will use the DNA Amplicon module. If we have RNA input, we will use the RNA Amplicon module. For DNA and RNA samples, we will need to use both modules separately. For more information and support for BaseSpace apps or LRM and its modules, please see these links. I'll now give a quick summary of our talk today. We discussed that Amplicon sequencing is a method that gives us a closer look at specific regions of interest in the genome. Amplicon libraries are lower in diversity than other library types, and some are very, very low in diversity. We discussed 3 methods to prepare Amplicon libraries: AmpliSeq for Illumina, Nextera and Nextera DNA Flex, and the Illumina demonstrated protocol, one application of which is the 16S metagenomics protocol. We reviewed Amplicon sequencing metrics, focusing on the percent base, a measurement of nucleotide diversity, clusters passing filter, which is a measure of the percentage of the clusters on the flow cell that gives pure signal and can be used to generate data. Q-scores, overall measurement of data quality. We discussed the 2 strategies that we want to use together to improve low diversity Amplicon runs. The first is spiking in phi-x, and the optimal spike in percentage will differ depending on the sequencer and chemistry you are using. The second is reducing the cluster density. We can analyze Amplicon data with BaseSpace or Local Run Manager, also known as LRM. We can use the DNA, RNA, or DNA plus RNA Amplicon apps on BaseSpace and use the DNA or RNA Amplicon modules on LRM. I have some additional resources for you as well. We have public data sets including 16S and AmpliSeq panels on BaseSpace, which are a good reference for the sequencing run metrics and sequencing analysis. We also have the Library Prep Kit Selector, which is helpful if you are unsure of the best method to use for your project. The Bulletin Board, which has helpful documents on a variety of topics. NGS for Beginners page, which has a variety of resources for getting started with NGS, also known as Next Generation Sequencing. And the main support page. We also have recorded online and upcoming webinars, and you can always contact technical support with any questions. Lastly, we have 2 upcoming library prep webinars. The first is Library QC and Troubleshooting with a Bioanalyzer. This webinar will discuss how to use the Bioanalyzer and Fragment Analyzer as a tool for library quality control and how to identify the features of an ideal final library trace. This webinar will be presented on February 25th, 2020 at 11 a.m. Pacific Time. The second webinar is Sequencing, How to Plan Your First Sequencing Project. This webinar will discuss the end-to-end workflow for planning your first sequencing project from idea to data analysis, including helpful resources along the way. This webinar will be presented on April 9th, 2020 at 11 a.m. Pacific Time. I can now take your questions if you type them into the chat.